Okay, so today we're going to talk about translation. So we've already talked about turning DNA into RNA with transcription, and now that RNA, we're going to talk about how that's going to be turned into a protein. Okay, so we have our copy of our gene. It's still as RNA, so it's actually a messenger RNA strand. That mRNA can leave the nucleus and be used to make a protein, and so this is in the form of RNA code. And it's determined by the way these 4 different nitrogenous bases are arranged in the DNA. So they can be interpreted into the amino acids for the protein, and that's how what translation is turning that protein, that RNA sequence into a protein. So this is going to be happening at the ribosome. So remember, the ribosomes are in the cytoplasm and also attached to that rough endoplasmic reticulum. Okay, and so the information is translated from RNA language of the nitrogenous bases to the language of proteins or amino acids. There are 20 amino acids and 64 different combinations of A, U, C, and G. So because of that, we can actually have multiple codons that code for the same, or multiple combinations of RNA that can code for the same amino acid. So they're read every 3 bases. This is going to be the ribosome going through and associating every 3 bases in the RNA with 1 amino acid. This set of 3 is called a codon. It's that 3-letter code word for the mRNA. So each codon along the mRNA strand corresponds to an amino acid, and we have ones that signify the start and the stop of translation as well. So there's charts like this. You might remember this from regular biology, where you look up the codon and it tells you the amino acid that you have. So you have first position is that U, C, A, or G. The second position, then you use that to figure out what column we're in. And the third position to figure out which one. And these 3 letters represent amino acids. Notice all 4 of these code for the same amino acid. So we can have multiple codons code for the same one. We also have ones that code for stop. Okay, so here's an example. So try this on your own. Okay, so, AUG, AUG would be MET, AGA, AGA would be ARG, CCG, CCG would be PRO, UUG, UUG, GLEU, ACA, ACA is THR, ACU, ACU, THR, and UAG, UAG, stop. So this is also how we know to stop making an mRNA or start making our protein as we get to a stop codon. You can also see these written in this circular manner, and we start at the inside. I actually like these a little better, but it's up to you. And I don't know what kind they're going to give you on the test, but you start in the middle and you work your way out. So first letter, second letter, third letter to give you what it is. Sometimes they'll have the whole name written out of the amino acid. Sometimes they'll use a 3-letter abbreviation, or there's even a 1-letter abbreviation. Okay, so ribosomes need 2 other things in order to change this mRNA message into a protein. They need rRNA, which we talked about a little bit with when we were talking about RNA before. That's ribosomal RNA, and it's helping make that bond between our amino acids. It's found inside the ribosome itself, which is made of 2 units, the small subunit and the large subunit. I always think these kind of look like little hands coming together with like thumbs, but so you never just see rRNA by itself. It's always going to be part of the ribosome. There's also proteins involved in the ribosome structure as well, and that's that large and small subunit. We also have tRNA. This stands for transfer RNA. This is how our amino acids, they're bonded to the transfer RNA, and it's how we bring the correct amino acid to the ribosome to create our protein. So it's going to bring the correct one. Yep. How does the tRNA know which codon to match up with? This is because of something called the anticodon. So here we can see that the tRNA actually makes kind of a T-shape structure, but we have parts that make little hairpins where they're bound together. Anyway, there's a section at the bottom where we have the anticodon, and what happens is as it comes into the ribosome, the anticodon can interact with that mRNA, and it can bind just like it would with DNA, where A goes with U goes with A, C goes with G, and G goes with U, and it would bond to its opposite. The anticodon would bond to the codon, and then that anticodon is going to be part of the tRNA that has an amino acid at this site up here at the top, and it can be added to our growing polypeptide. So here's kind of showing again, here's our mRNA. This would be all happening inside of a ribosome. Here is the codon, GAG. Again, when we use our charts to figure out what amino acid to use, we're always looking at a codon, okay? So the only real time we need the tRNA is for understanding how, or the anticodon is for when we're understanding how the tRNA works. But codon is what we look up on the chart. The anticodon, the opposite of that, attaches, and it happens to have the correct amino acid attached to it. Here is showing what's happening. The tRNA comes in, bonds to its opposite complementary strand, and then it attaches the correct amino acid as we go. Okay, there are 3 different sites. We have the first site is called the A site. It's where it arrives, the tRNA arrives. That's this site here. We kind of go backwards, right to left. So this first site is the A, the arrival site. The P site here in the middle, this is where that peptide bond is formed. So you can see that this bond is formed in where that growing peptide chain is added to the next amino acid. The last is E for exit, and as soon as it goes in there, the tRNA releases and floats off into the cytoplasm. So here's our mRNA strand again. The ribosome, codons down here, the tRNAs, the amino acids. And we've mentioned this before, but there are many different different tRNA. So typically there should be 61 different types of tRNA because there's 61 anticodons. But because it's often redundancy, the base pairing rules aren't as strict on that last one, which is kind of why you might notice on our charts, let's go back really fast. Oftentimes like A, C are very strict, but any of these last 3 could make for threonine. So we can have the same one be used for a couple of different codons. Another thing you might notice is that this stop codon doesn't have an actual tRNA that attaches to it. So there isn't a stop tRNA. It just signifies to the ribosome that it's time to stop.